I'd like to thank the organizers of the Cancer Genomics Consortium Virtual Meeting 2021 for inviting me to give this talk. I'll be talking to you about artificial intelligence applications in precision oncology clinical trials. All the work that I will be presenting today were conducted at the Georgetown University's Innovation Center for Biomedical Informatics. So here's a quick outline of my talk. I'd like to present two case studies for the applications of machine learning and AI in precision oncology. The first one will highlight the usage of virtual molecular tumor boards. And the second one will talk about predicting toxicity in patients receiving cancer immunotherapies. And then after that, I would proceed to describe the Georgetown database of cancer, where the data sets used in the first two case studies are organized and available for larger use by the research community. And then finally share some thoughts about the applications of data science and technology in precision oncology. So I'd like to start the first example with a case study. This gentleman was a 52-year-old Caucasian male. He was diagnosed with stage 4 pancreatic adenocarcinoma and had possible lung metastasis. He was treated at the Georgetown University Lombardi Cancer Center. And this patient was progressing after multiple lines of chemotherapy, gemcitabine, Abraxane, 5-FU, nab-paclitaxel, et cetera. And later on, there was an addition of erlotinib, which is a targeted therapy. And he was continuing to progress on this targeted treatment as well. It was at that point that the oncology team decided to conduct a multi-gene panel testing of the tumor biopsy of this patient. And from that analysis, it was identified that this patient had a very unique mutation in KDR, which is a VEGF receptor. And additional curation and informatic review revealed that this mutation could potentially be pathogenic. So here's the quick biology of the VEGF receptor. Because of this mutation in VEGFR2, there was ligand-independent constitutive phosphorylation that led to this pathway being on all the time, resulting in angiogenic activity within the tumor cells. There are multiple tyrosine kinase inhibitors that block this anti-VEGF activity, and many of them were in clinical trials at that time. This study happened in 2017, 2018, and many of these TKIs were in clinical trials at that time. And because of this bioinformatic analysis of this pathogenic variant, a very specific NCI-sponsored sorafenib trial was identified, and this gentleman was eligible for that particular clinical trial, and he was matched and enrolled onto that clinical trial program. After a few months of being on the clinical trial, the patient sent an email to our patient coordinator saying that I'm working again, I'm traveling, I'm playing squash, and I've gained weight, right, which is a big deal for these patients with advanced metastatic cancer, pancreatic cancers. So that was a big success story for precision oncology in PDACs. The question then became, how do we scale this case study to every patient that's diagnosed with cancer? And so obviously we cannot do this manual review and manual curation of data. Can we really deploy data science and technological methods to scale this process to all patients diagnosed with cancer? And so to answer that question, we built an artificial intelligence-based virtual molecular tumor board. And the primary goal of this tumor board was to organize the data, such as the molecular variants data, drug approvals from the FDA, clinical trials from ct.gov, and other useful information such as data from electronic medical records for patients into a virtual tumor board knowledge base. Once this data was organized in the virtual knowledge base, then filtering and prioritization was applied on a patient-by-patient basis, and the results were presented to a medical review panel. And the medical review panel members would then review the results and make a determination as to which trial was appropriate for this patient. And here on the right-hand side, I've shown some of the data sets that went into this knowledge base, which included clinical data, molecular profile of the patient, and also the results of outcomes that were collected for each patient went back into the knowledge base so that they could be helpful for future patients who were analyzed using the virtual tumor board. This program was sponsored by PanCAN, the Pancreatic Cancer Advocacy Network. So every patient that came through the Know Your Tumor program within PanCAN was registered into this IRB-approved observational study, and we were able to do this analysis and provide both a physician report and a patient report to that patient of PanCAN. So using this methodology, we were able to scale the availability of precision medicine to patients across the United States from over 300 hospitals in a short time period of between 2 to 3 years. So here are some quick results from the Know Your Tumor program. Multiple molecular testing was done on these patients to match them to appropriate therapies, next generation sequencing analysis, chemopredictive biomarkers were analyzed using immunohistochemistry data. And for a very select set of patients, RPPA was conducted, which is the reverse phase protein arrays, to really understand phosphorylation of certain key proteins. And this was done only in a small number of patients as the assay itself was being piloted through the Know Your Tumor program. This graph here shows basically how the patient's progression-free survival was when they were treated with therapies based on their molecular markers, right? So in the case of this first patient, he or she received therapy based on their molecular profile. They were HER2 positive, and therefore trastuzumab was added to their chemotherapy regimen. This third patient, for example, had a BRCA2 mutation, and therefore olaparib was added as a treatment option for this patient. Here in the x-axis, we see progression-free survival, and this is rank ordered by patients who received matched therapy and how their progression-free survival was much longer than some of the other patients who didn't have as many actionable biomarkers or did not receive therapies that matched their molecular profile. The different colors here indicate the treatments that these patients had prior to coming into the Know Your Tumor program. They might have had prior platinum therapy, or they had standard of care, or were on another clinical trial, or they had, like the case study that I just presented, they had multiple lines of therapy, 2 or 3 lines of therapy before entering into the Know Your Tumor study. And this was the final result of the Know Your Tumor program, where over 1,000 patients were enrolled. What we found was that patients who had highly actionable biomarkers and received matched therapy almost doubled their progression-free survival compared to those patients that had highly actionable biomarkers but did not receive a matched therapy. Of course, this was an observational study, and this has to be validated through a randomized control trial, but it gave a lot of pointers for designing the appropriate randomized control studies for these biomarker targeting agents. So we went from one patient to how do we scale this across the United States? And then the question became, can we create global collaborations that will enable this beyond just the tertiary care academic facilities or developed countries? In order to do that, we quickly recognized that, the hospitals themselves may not have all the expertise that are needed to do this kind of genotype phenotype matching and a tumor board analysis to be able to provide recommendations to their oncologists or their patients. So this mechanism was proposed, and you can read about this more in JCO-CCI, which was published last year. This concept of a multi-institutional expert panels. So essentially what this entails is that as a clinician sees a patient and he or she recognizes that we need a virtual tumor board, then de-identified version of those patient records could be sent to this multi-institutional expert panel, and analysis would be done in this multi-institutional tumor board, and results and recommendations would be presented back to the clinician. This is very much in a conceptual stage, and you can imagine the need for highly validated and verified databases that can power this type of analysis, and open databases essentially that are curated by experts around the country, around the world, based on the types of patients that they're seeing. So that leads us to, how do we organize this information to power these types of global expert panels? And it literally takes a village to organize these processes and databases and tools. I'm showing you one example of how we organize this data. This effort is funded by the Genome Institute. It's called the ClinGen program. Within the ClinGen program, there's the Somatic CDWG, or the Clinical Domain Working Group, which focuses primarily on somatic variants that may be associated with treatments. And this group has been meeting since 2015, and we curate this data using something called as the MVLD, the minimal variant level data. So these pretty much are the standards for how do we represent a somatic variant for an individual patient. So the case study that I just presented to you, this patient had a VEGF receptor mutation, and that was correlated to one of the TKIs. So what are the basic set of data elements that we need to capture for each of these patients to represent not only their molecular profile, but also the therapy indication that might be associated with this mutation profile. So we described that using a standard called MVLD, or minimal variant level data. This was published a few years ago. And I just show you a schema of MVLD here. Won't go into the details of every data element, but primarily this framework is divided into three parts: allele descriptive, which essentially tells you what exactly the mutation was, and the allele interpretive tells you whether or not there were protein changes because of this gene variant. And then the somatic interpretive tells you whether or not it's a diagnostic variant, or a prognostic, or a predictive, and what were the class of drugs that are associated, that might be associated with this particular variant. As soon as MVLD was published, soon after that, the AMP guidelines came out. The AMP guidelines really provided a framework for how we should think about somatic variants in terms of different levels of evidence. And there's an uptake of this AMP guidelines by the FDA as well, in terms of how these variants are reviewed for precision oncology treatments. And so we took MVLD and we integrated that with the AMP guidelines, so that the MVLD guidelines now look like this. So most of the somatic interpretation now happens using the AMP guidelines, which is shown here. So in terms of the teams themselves, the CDWGs, or the Somatic Clinical Domain Working Groups, are divided into multiple task forces. These disease-based task forces are the ones that are organizing these expert panels that I mentioned before. These expert panels could be based on a pathway or a particular gene. So for example, there could be an expert panel entirely based on NTRK fusions in pediatric cancers. Or they could be based on FLT3 mutations in hematologic cancers, or FGFR mutations in bladder cancers, for example. So these task forces then spin off these expert panels to allow for curation of these variants, and also additional evidence that support associating these variants with various therapy indications. Where do we put all this data? We partnered with the Civic platform, led by Obi and Malachi Griffith at the Washington University in St. Louis. That is the database that holds all of the variant curation that these expert panels conduct. And the curation in Civic is guided by the MVLD framework, the minimal variant level data, which is now integrated with the AMP guidelines. The Civic platform itself, once the crowdsourcing and the curation happens, this data can then be automatically submitted into ClinVar, which is really the database of record for clinical variants. And there are many users, both in academia and the public sector and private sector, that use the ClinVar database. And so all the data that we curate enters into the ClinVar database so that it's more broadly available to the biomedical research community. So here is a quick screenshot of the curation teams. Within the civic platform, there are different organizations. So one of the organizations is the pediatric task force, for example. So everything they curate kind of goes into that bucket. And like I mentioned, there is a whole process of curation, review, adjudication, and sign off for the somatic variants that enter into the database. This civic database is also contributing to global efforts such as GA4GH, the Global Alliance for Genomic Health, which is integrating somatic variant data from multiple different data sources. And each of these databases might have certain area of expertise or deeper annotations in certain areas like adult tumors or pediatric tumors or certain pathways or genes. By bringing these databases together, we then allow the users to be able to see annotations wherever they were available across these 6 different databases. So bringing us back to this case study, this N of 1 case study that I presented at the beginning of my talk. So how do we kind of take these processes and tools and methodologies to really scale the quality of life improvement and the appropriate matching of clinical trial that this patient has to every patient that's diagnosed with pancreatic cancers or other types of advanced metastatic cancers. And definitely the ClinGen Somatic Working Group is working very hard to make that happen at a global scale. The second case study I wanted to present to you all today was predicting toxicity in cancer patients who receive cancer immunotherapies. The ICIs or immune checkpoint inhibitors have really transformed outcomes for a number of cancer patients. These immune checkpoint inhibitors, they modify the patient's immune system so that they become much more aware of the cancer cells, and they're able to destroy the cancer cells because of this heightened immune system, because they were given these immune checkpoint inhibitors. Immune checkpoint inhibitors are really catching on. They're now given to patients with multiple cancer types, melanoma, kidney cancers, lung cancers, etc. As these drugs were being rolled out, there are multiple registries that are beginning to capture the treatment-related adverse events. I mean, no cancer treatment comes without any adverse events. Most of these drugs have adverse events. The only question is which ones are really serious and which ones are mild. If there are serious adverse events, how do we manage them? If the drug is really helping the patient, then you want to keep them on that particular treatment to really shrink their tumors, but at the same time also manage the adverse events so that they can stay on that particular treatment. So the question is, can we apply machine learning methods to really detect who are the patients that have these adverse events, so that once we understand these features, we can then predict in the future if patients are receiving immune checkpoint inhibitors, who are the ones that are going to end up with serious adverse events. So that was the question we asked in terms of mining the patient charts to detect immune-related adverse events. It's an extremely time-consuming activity. A lot of the chart reading has to happen. It's costly and lacks standardization. So the question is, can we apply in natural language processing techniques to not only extract, but also standardize immune-related adverse event data? So here I'm going to tell you a little bit about the registry that was constructed at the Lombardi Cancer Center. And now it has grown to over 1,000 patients. These were patients who were receiving immune checkpoint inhibitors. They were diagnosed with a variety of cancers, bladder, GI cancers, head and neck tumors, etc. And these were the different types of immune checkpoint inhibitors. They were either anti-PD-1, anti-PD-L1, or anti-CTLA-4 antibodies that were given to this patient. And the registry itself was created by conducting a retrospective chart review of the patients that were entered into the EHR. And so here's a quick schema or architecture for how the registry was constructed. There were multiple source databases. We had the pharmacy data with the drug information. We had the registry data, which was data from the electronic medical records, and additional curation by the residents. And then there were a lot of additional clinical data that was gleaned from the ARIA EHR database. All of these were extracted, transformed, and load processes were applied to them, and the data was loaded into the REDCap database. Once the data is in the REDCap database, it allows for, it has multiple dashboarding tools, data exploration tools, and I'll show you some examples of this. Here's a quick demographics dashboard, which pulls data from this REDCap registry. You can do some quick visualizations, such as for, vital status or gender, race, toxicities, who had toxicities and who did not. Were there additional comorbidities in these patients? Did they have any molecular profiling data that was available? So that's what is indicated by this CADIS platform here. An additional analysis could be done to see what types of adverse events were present in these patients within the registry. We could also subset the data based on the comorbidities that were present in this patient population. So what were the NLP tasks to extract this information into the registry? We automated the identification of patients using ML approaches. The input really was longitudinal clinical notes for patients treated with ICIs. And the output was identification of patients who developed immune-related adverse events. So here's an example of a clinical note. So for example, this patient here was using ipilimumab. That was a drug that was given to them. And basically the clinical note said that this patient was receiving ipilimumab, had ipilimumab-induced diarrhea. And so there is a possibility that they were developing colitis, which is one of the serious adverse events for immune checkpoint inhibitors. Right, and then the word diarrhea was mentioned multiple times in the clinical notes. So here's a quick methodology processing, natural language processing methodology. So the registry that I just described to you, that plus the clinical notes were used for textual processing. There was a lot of note filtering, et cetera, because there were subsections within the clinical notes that had to be sectioned out and separated before the NLP tools could be applied. And then feature engineering was applied to prioritize the features that would help us select these immune-related adverse events. And then finally, these features were used as an input into classification models, and the classification models would output whether or not the patient had an immune-related adverse event. So we processed about, close to 10,000 clinical notes that were extracted from the Georgetown Lombardi EHR system. They were de-identified using a tool called DID. It removes all the 18 HIPAA identifiers. And once the de-identification happened, then the different section headers were parsed out, based on whether it was a medical oncology note, whether it was a nurse note, whether it was a progress note, et cetera. And a variety of filtering techniques were applied to reduce noise. For example, we applied things like heuristics where the patient denies rash, right? So it's actually a negation sentence. So the patient did not have rash. So if we just did an entity detection, then you would detect rash and say maybe this patient had rash, but you have to look at the neighboring words to understand whether it was a positive assertion or a negative assertion. So things like that were done to filter these nodes and to extract the nodes that were most appropriate to feed into the classification model. Two types of machine learning methods were applied: classical machine learning, logistic regression, support vector machine, and random forest. And we also applied deep learning methods, convolutional neural network, and also BiLSTM were two deep learning methods that were applied to the feature set that was extracted from the clinical notes. And we performed tenfold cross-validation to evaluate the classification models. Even though this was all part of the Georgetown registry, the data actually came from 10 different hospitals that participate in the immuno-oncology program. So we had a good heterogeneity of patients from across the greater Washington, DC area. Here are the results from the analysis. The results showed that the CNN, or the deep learning method, had the highest precision and recall, and also the F1 score compared to some of the classic machine learning methods. Even this has a long way to go. The recall is not exactly where we want it to be. Really, this was just notes from about 1,000 patients, and increasing that number would help us increase the precision and recall on this, so that we could have a larger number of training data for the machine learning method. So far we've talked about two use cases or case studies. One is a virtual tumor board, which involved a lot of data integration across genomic and clinical databases, and how do we apply machine learning methods there to prioritize clinical trials that the patient may be eligible for, and presenting that to clinicians. And in the second example, we talked about a natural language processing based methodology that would help us rapidly detect patients who have treatment-related adverse events, so that they could be identified for various clinical trials involving immune checkpoint inhibitors. And both were supporting clinical decision-making. First one more for cancer care, and the second one was more towards identifying patients for appropriate clinical trials. Now, we've talked about a lot of data sets, data harmonization is required across these multiple databases. And that was the goal behind the Georgetown Database of Cancer. And we've developed this over the past decade or so. This is really the translational medicine database of cancer at Georgetown University. And multiple studies are entered into the Georgetown Database of Cancer and are utilized for a variety of translational research purposes. Here is one example data set in GDoc, and I just wanted to show you the life cycle of it. This is a brain tumor data set that originally was initiated at the National Cancer Institute with 14 different sites in the US. This was primarily for brain tumors and for genotype-phenotype integration for patients with glioblastoma multiforme, oligodendroglioma, and other types of brain tumors. And this database and the Rembrandt data set was noticed by President Biden, who made a call out for GDoc and Rembrandt on Twitter. So here's a quick schema and an architecture diagram that shows not only the data sets, but also the types of data that are supported in the bioinformatic pipelines. Just in the last year, we have undergone a big facelift for GDoc. Now it's not a centralized database with tools sitting on top of it, but rather a collection of tools that both developed at Georgetown and also developed at other places like Johns Hopkins and University of California, bringing those state-of-the-art tools to the data sets that are sitting in GDoc has been the primary goal of the second generation of this software platform. So very quickly, this is the impact that a platform like this has had. We had multiple cancer types, about 61 high-quality data sets. I only mentioned Rembrandt, but there are many other data sets within the GDoc system. And there are over 3,000 researchers from across 48 different countries that use this platform for a variety of research purposes. It has really become the basis of bioinformatics education at Georgetown. So we developed this course called Demystifying Biological Big Data, and it's entirely based on the GDoc platform. And this had over 4,000 students within a span of 8 months that participated in this MOOC, the Massive Open Online Curriculum, and it has contributed to a number of publications across different cancer types. The platform and the Rembrandt dataset was also used by the FDA for their Precision FDA Data Challenge, and this was a data science challenge that was announced in 2019 with 3 winners who were announced a few months later. And really the goal of this was to apply machine learning methods to predict outcomes in patients with brain tumors based on the biomarker data. So it was a fun data science challenge, and the GDoc platform and the Rembrandt provided the basis for this GDoc, for this Precision FDA challenge. So finally, I'd like to just leave you with a few final thoughts here. These virtual tumor boards are really catching on, especially what COVID has taught us is that We need these types of virtual platforms to really conduct, both cancer care as well as cancer clinical trials. And where the pharmaceutical companies are moving is really, can we bring these trials to patients, right, almost in real time, so that we eliminate that startup time, et cetera. And these virtual technologies and digital technologies are going to be critical in enabling that. We also showed that AI tools like the NLP tool that I showed you to detect patients with adverse events are going to work alongside clinicians and researchers to support treatment planning and also enrollment of patients into clinical trials. And we'll see more and more of these distributed types of data lakes or data ecosystems that capture multimodal data and really enable users to connect information to answer business questions, be it in an academic center or in a pharmaceutical company or in a biotech organization, so to say. Just very quickly wanted to put this up. We at Georgetown, there is a one-year master's program in health informatics and data science. Many of the researchers who conducted all the work that I just presented to you teach in this program as well. So if this is an area that you want to get into, take your lab expertise and apply more data science to it, this might be a program for you. You can look that up at healthinformatics.georgetown.edu. Just want to thank all the collaborators who participated both in the virtual tumor board program and also in the ClinGen Somatic CDWG, or the Clinical Domain Working Group, and the funding from the NIH and the NHGRI. And for the virtual tumor board, the funding came from PanCan, the Pancreatic Cancer Advocacy Network. And this is the core team at Georgetown that developed the Georgetown Database of Cancer. Thank you very much.